Hi, I'm Lisa and I'm trying to build smarter microscopes. Cells are incredibly dynamic. They move, they can sense and respond to their environment. A classic example is wound healing. When we get a cut, growth factors are released from the wound site and cells can sense this gradient and travel towards the wound site like a dog sniffing a scent trail. In the Pertz lab at the University of Bern, we study how cells can process these signals using optogenetics and genetically encoded biosensors. Optogenetics let us control cellular functions simply by shining light onto them. Using a microprojector, we can shine light onto tiny regions, even subcellular ones, and using the biosensors, we can read out the cellular responses. This means we can, for example, create artificial growth factor gradients and make cells move into a specific direction, simply by shining light, like a cat following a laser pointer. These experiments showed striking heterogeneity. While most of the cells traveled towards the growth factor gradient, some cells did exactly the opposite and traveled away. To understand this kind of heterogeneity, we want to study those processes with high throughput. This is why I built a robot that can automatically shine light onto hundreds of cells in parallel, so we don't have to do it manually. This smart microscope gives us unprecedented control over cellular behavior. We could even reprogram swarms of cells to create specific shapes or letters like a biological tissue printer. To better understand how cells process spatial signals, we wanted to homogenize their geometry. Using micropatterning, we can stamp adhesive proteins onto glass slides to make cells grow into specific shapes. I developed a method to convert existing microscopes into 3D printers, allowing us to rapidly prototype stamps and microfluidic chips in-house, cutting down cost and iteration times from weeks to days. With this technique, we can now test how cells respond to spatial input across hundreds of identically shaped cells, experimentally confirming long-standing mathematical theories. To allow the robot to see where to shine the light, I also developed new computer vision tools that allow users to simply teach the microscope to recognize certain structures in 2D, 3D, and even time-lapse movies. The tool worked so well that we released it as a standalone tool and it's now used by labs across the world for different image analysis tasks. So what's next? With funding from SNF, I'm trying to build what we call the ChatGPT microscope. It's a natural language interface where users can simply describe the experiments and an LLM plans and executes the experiment on the microscope autonomously. In the future, we aim to implement robotic-style task planning to advance the automation of microscopy-based biological research. Finally, a big part of my PhD has also been to share my excitement for biological research with the public through social media, public events, but also collaborations with musicians and visual artists. I believe we scientists have a responsibility to make our work accessible and engaging. When communication breaks down, we not only risk funding cuts, but public mistrust can also lead to huge ecological and public health crises. We spend months writing papers for a narrow audience of specialists, but often forget to also explain our research in a way that everyone can understand. And I think this is something that we really need to change. I hope this gave you a glimpse into my PhD journey at the Pertz lab and also made you a little bit excited about the smart microscopes of today and the future. Thank you.